Hi everyone, welcome to UCLA. My name is Siavash Kurdistani. I am a faculty member in the Department of Biological Chemistry, and my lab is in the Biomedical Sciences Research Building. We have recently discovered that histones function as enzymes in reduction of copper, so they're copper reductases, and that is the main focus of my lab. This finding stemmed from the broader question of why eukaryotes have histones. The typical answer that you might find in a textbook is that histones are involved in genome compaction, and also histones regulate essentially all processes that are based on DNA, including regulation of gene expression. But it was unclear to us whether these were the functions for which histones evolved in the first place. This is because ancestral histones, which form a structure very similar to the H3H4 tetramer, shown here in the mustard color, which is H3, and the blue color, which is H4, occurred in organisms with small genomes and little capability for epigenetic gene regulation, suggesting that histones may have had or may still do have an older function that is conserved in today's eukaryotes. Following this logic, we discovered that the histone H3H4 tetramer is an oxidoreductase enzyme that reduces copper inside the cell from a 2+ state to a +1 state. This finding has been reported just recently in Science, and so I refer you to this paper to learn more about the details of this project. The reason this reaction is important is because copper 1 is the biousable form of copper. It's the form that's trafficked inside the cell and transported to the destination proteins in mitochondria and in the cytoplasm. So places that copper functions include electron transport chain, so you need copper for respiration, as well as important enzymes such as SOD1, which is involved in oxidation defense. We believe the catalytic site is at the interface of the two H3 proteins, with important residues contributing to binding of copper and transfer of electron to it to reduce it to a copper +1 state. Our current and future projects are focused mainly on the implications of this new discovery. We're interested in understanding the mechanism of catalysis, looking at a structure-function relationship of the histone H3H4 tetramer. We would like to understand how this enzyme activity is regulated, specifically through post-translational modifications of histones, as well as histone variants, including variants of H3 in the context of H3H4 tetramer, as well as the variants of other histones in the context of the nucleosome. We're performing CRISPR-based genetic screens to identify the molecular factors and pathways that link the new histone enzyme activity to the broader copper homeostatic network of the cell. And importantly, we're interested in linking the histone enzyme activity to human disease. We're focused on two classes of diseases. One are diseases that involve the dysfunction of iron sulfur clusters. These are cofactors that are produced in the mitochondria and are required for proteins that are involved in essentially all metabolic and pathways, including pathways that are involved with DNA function. Many of these diseases are neurodegenerative in nature, and in fact we have quite a bit of evidence showing that histones are involved in pathogenesis of at least one of these diseases. Histones are also mutated in certain cancers quite a bit. For example, in lung cancer, up to 40% of patients carry mutations in one of the histone genes. And we're obviously interested in understanding how these mutations potentially affect the histone enzyme activity and contribute to the cancer phenotype. Thank you.